Hi, everybody.
Hi,
Hi, Lisa Tay.
Oh, good afternoon.
good afternoon.
Welcome, welcome. Some folks are still connected. So, Brianna, are you running this?
I believe so. So welcome in, everyone. Welcome to the second Chat with a Scientist of this year. I believe we're going to do something similar to what we did last time, but we're mainly going to be focusing on your research proposal questions. So if anyone's got any questions, feel free to either unmute and say them out loud or even put them in the chat as well. And yeah, so we can go ahead and get started.
Okay, well, Trent and I maybe could do a little spiel to kick us off, and then people could raise hands. So, we're coming to the time when you want to do your independent research proposals. So you've been doing your standard trials, which are super important to us because those are all the data that we're really excited to get and to use. But your research proposals are really where you guys get to be creative. And, that's really exciting and a lot of fun, right? So there are a lot of things that you've been thinking about, a lot of things that you've been measuring, things that you've been testing out. And the research proposal gives you the opportunity to take that one step further, but to do it in a way that's completely unique to you and your team. And so, for instance, if you are doing one of the experiments with light, in the past, we've had a number of people who've then gone the next step to look at different light settings for the plants to compare with the light settings that you do in the primary trial and see, well, how do the plants grow different with different colors of light, different durations of light, different intensities of light. So all of those different settings would be the independent variables. These are the things that you are intentionally changing in your experiment to see what happens. And then the see what happens, that's your dependent variable. That's what you measure, whether it's your plant biomass to compare the biomass that you grew before, or your plant height to compare to the height from the plants that you grew with the other settings. So those are the types of things that you could change, but you can change all sorts of different things. You could change different types of plants, different types of nutrients, different types of, all of those cardinal factors that plants need to grow. Light, temperature, nutrients, moisture, atmosphere, if you can come up with a way to do it, and other things, any other aspects of the experiment can be changed. But the key thing, the really important thing, is that you can only change one thing. Because if you change more than one thing, you have confounding situation. You have confounding variables, and you don't know if you see a difference in plant growth, what that difference is due to if you have more than one independent variable that you're changing. So that's one of the really critical things when you're writing this proposal. And it's one of the things that we often see, people make mistakes in this because they try to change too many things. And if you change too many things, you just can't compare anymore. You have to do more experiments to be able to do that. And maybe that's something you want to consider too. Maybe you want to do short experiments so you could change a couple of different things. All those are possibilities that be on the table that you could put into the proposal when you're thinking of writing it. So, Trent, what did I miss?
I was going to say, just be careful if you're going to shorten the experiment to be sure that what you're measuring is something that you get enough signal that it's meaningful, right? So, for instance, if you're trying to check fertilizer, the plants don't really start using fertilizer until they're through the microgreen stage, right? And that's when they hit their exponential growth and start adding a lot of leaves and leaf area. And
About
so,
2 weeks
if I'm checking fertilizer, I really got to grow it out at least through the exponential growth phase, right? So, because otherwise, I'm not really testing the fertilizer because most of the energy is coming from the seed. So
Exactly.
we need to think through the experiment to make sure that the signal that I'm measuring, the thing that I'm measuring, it makes sense and is meaningful. So that's the other thing to also consider.
Yeah. So it can be pretty simple. It can be a little more complex, but
I
yeah.
love simple. I got to tell you, because you've run some really, really important experiments. These are critical. We don't know the answer to the questions that we've asked, right? This is why you're running the experiment. The answer is not in the back of the book. We're going to look at the data in aggregate and see what we see. And now that you've run your experiment, and you're like, wow, this was cool, or whatever you think. I always think it's cool, but that's me. Wow, I wonder if we change this, what might happen. I think this might happen, right? So that's your hypothesis. So we always, any experiment I've ever ran, I never answered the question totally. Typically, we answer the question, but we would see some other things like, wow, that was interesting. I wonder what would happen if we did this. I think this might happen, right? So this is the cool thing in science, is we always have more questions than answers, it seems.
And can I just add one thing that I think, so as you know, in Growing Beyond Earth, there are 2 trials, right? So there's the first trial where you are all following the research that Joya and Trent are very interested in the answers on. So you're following their protocols. For the second trial, the idea is for you to analyze what you've learned and try to figure out a way that you can test the things that make you curious. Like, what is it that makes you curious? What is something that happened that you're like, I don't know why this happened. Maybe we can figure out why this happened. You change that variable, maybe that'll help you. But really thinking about the thing that makes you curious, because what happens is that the original research that you guys conduct is the stuff that brings new ideas to NASA research. So your ideas are new ideas. And in the past, we've had new ideas about like continuous harvest that came from students through this program from their original research that allowed the NASA folks to test that on the International Space Station. So maybe you'll come up with some innovative challenge or something that we haven't even thought of a way to test. And maybe you'll come up with that, and maybe that will be used by NASA. And then the other piece of this is not only do you get to do the original research, but then you get to present it to a bunch of NASA scientists if you want at the end of the year in April. So once you guys are thinking about all of this research and you're coming up with your proposal and you're thinking about what that new research is going to be, also think about how do you, as you're going, you're thinking about how you would present that. And we'll talk about that at a later time, but you'll have the opportunity to present that. And then it might be something that's usable by NASA for them on their next mission or their next experiment that they send.
Right, or it might be the next Growing Beyond Earth project that we roll out to a lot of different schools too. So that's another thing that we may do to even get more data on that topic. That's a great point. And yeah, I think thinking about the symposium and thinking about, like Amy said, what's an interesting question? What's a topic that's interesting? Do a little bit of research. Like read up on things if it's something that's of interest, even before you write the proposal, if you can, say that you're having, well, I see germination issues in the chat, and think about, well, what factors could be going on with the germination? Well, maybe it's the moisture content, so you could read up a little bit about substrate and substrate moisture and how it may differ. And then you can imagine, well, how could we change this variable and what could we do? Maybe those plants could be grown in a deeper tray with more water in the bottom, or, those substrates, or maybe you could use some sort of wicking material, so think about those types of things, read up on what people have found out about substrates and moisture, and that may spark some ideas if there's a topic that's of interest. And I'm not sure, Brianna, if we want to answer that question too.
And also, Brianna, do you want to, when we get to a point, you want to share, like, just reiterate how you go about writing the proposal and where the data and all that, I mean, where that goes to and all that?
Yeah, for sure. So, let me see. So writing your proposal itself and submitting it, we are going to be using a Google form this year. So we have, I believe we have sent out that Google form. If not, it will be coming out shortly. but pretty much you're going to be able to fill out each of your variables, a little bit of background, and why you also want to be doing that experiment and what kind of inspired you guys to choose that variable to test and what and what you'll be testing. And then once you fill out that form, it comes to us, and then we'll be reviewing it with Joy and Trent over here. And then for that second trial, you'll be able to use your same consumables that you got from the first trial to carry out those experiments and see what might be what cool results you might get.
Or no,
But
Kylie, I
yeah,
don't know.
if you guys have anything to add to that.
I would
But-
just say
should
that,
just
you know,
knock
we're gonna review all the proposals. We'll give you feedback. That's the idea. So, you know, everybody needs a little feedback. So you might get some feedback. You might, things like you might consider doing this, or you might consider this. And so having these experts look at your proposal will allow you to get a little feedback to either narrow or expand, or maybe it's just perfect the way it is, or maybe there's something bigger that we suggest that you change, but we're gonna hopefully just give you a little bit of feedback that makes your second experiment even more successful.
All right. And with that, we can go ahead and get started with some of our questions. So we've already got one in the chat regarding the first experiment. Let me see. Our vermiculite and perlite seeds did not germinate. We planted 8 days ago. Should we abort the first trial, or should we keep growing the arsolite plants, which all germinated? Which will be our first question. I mean, for the first trial, you definitely can keep growing those plants. But that can also help you guys kind of think about what you'd like to test in the second trial and what maybe why those plants didn't germinate in the perlite and vermiculite, or maybe some other types of media and whatnot to see which what helps there. But
Yeah, I was going to say if you keep the plants growing, you can at least see they're 8 days old. So, and if you know, probably 8 more days, they'll start hitting exponential growth, and so you'll be able to see how they do, and that might help you write the follow-on proposal should you choose to do so. So I'm always a proponent of keeping experiments going when the plants are alive. Of course, you know, if they're dead, that's a different question.
Yeah, and I would also agree with Trent in the fact that we still need that data. Like, so you're growing those plants, and if those plants are alive, just keep growing that. I mean, it's the data it's the experiment, but it's also the data that's most important. So remember,
Yeah.
make sure that you're adding your data into the data portal, because that's super valuable, and making sure you're adding your notes. Maybe there was something that you noticed in that pot that may have contributed to why they didn't germinate. So all of your notes are critical to determine it, like to helping decide what is working and what's not working, because all of these experiments are important and they're valuable, and every piece of data, whether that be a number, a measurement, or some note that you wrote, that's all super valuable to the final product.
Yeah, and it's good for us to learn how the vermiculite's working for everybody. You know, we're looking at potentially changing out some of the media that we use for flight experiments due to the issues that we've had with the natural product with the arsolite. So the more data that you all can provide to us on how it's performing, things that are going wrong, things that are going well, you know, both are really important. So please feel free to you know, keep growing in the arsolite, provide those observations in the notes, and that would potentially give us clues on what to do. I don't know, Joy, what do you think?
Totally, yeah. I mean, non-germinated, that's a good data point. So it's a bummer because obviously you want all your plants to grow, but you know, unfortunately, science and space science doesn't work that way. And we've had many plants that didn't germinate in spaceflight, but again, it's good data. You know, at some point, maybe at the end of the experiment, you could kind of do some digging in there. You can pull the seeds out. You can see, did they even start to put out a root? You know, and so you might be able to get some information by trying to do some little failure analysis to try to understand the failure. But, you know, even with them not germinating, it's good to leave the pots in place. Maybe you could pull the seeds out at some point, but leave the pots in place because that impacts the other plants in your chamber as well. So you know, you don't really want to change things in midstream. We need to collect all the data.
All right. So for our next question, it comes from James Owena, who says, in the special media trial, we have 3 plants per pot, and at this state, they are so much tangled together. Can you explain the best way to do the measurements?
So did we not have thinning as part of that special media trial? I can't remember the
Yes,
protocol.
so we do have thinning, but it's usually from 8 seeds to the 3 seeds.
I see.
One thing you could always do is take out the plants and try and separate them as best you can to do the measurements. But
Well, just for the
not
final
sure.
harvest, right?
Yeah, for the final
Yeah.
harvest, yeah.
Yeah, I think at this point, you know, just do them as best you can and then put in the notes what you did. So try to be consistent. Like if you're just going to do the height and the area, you know, weekly per pot. I can't remember, Brianna, if that was, were they supposed to do each individual plant for height and area, or were they doing the whole pot?
It's an average for each pot. So
Yeah, so then you can just, you know, group the plants together. But like you said at the end, I think when you cut them, you can try to cut them and sort of really gently. I mean, the leaves get tangled, but you can just sort of gently tug them apart to try to get the individual sizes. Does that answer the question? It's tricky.
Yeah, I think it answers the question, because we have been trying to just measure, you know, the whole pot, because it's really not easy to measure them individually.
Yeah, I think that should be fine. And just put the notes in what you do.
I was just gonna say pipe in,
Yeah.
yeah, just make sure it's in the notes so we know what was measured.
Oh, yeah, we'll do that.
But that is a good note, maybe that we should think about for the future, Brianna, when we, you know, do the next round of protocols. You know, maybe there'll be some easier ways to do the pots with multiple plants. So it might
Yeah.
be something we want to improve on for the future. So thank you, James, for the comment.
all right, our next question is going to be from Evelyn, who asks, we are part of the light versus radishes challenge. Initially, we had a light combination of 144 red light, 72 green light, and 72 blue light. We found information that 3/4 of the light should be red and 1/4 of the light should be blue for better plant growth. We would like to know if we should change our light settings to 144 red and 48 blue. Would this correspond to 3/4 red and 1/4 blue? Let me see. I think for this trial, I would definitely keep it as the original light recipe, but it's definitely a good idea for your second trial experiment to try and see if you can test out that 3/4 red and 1/4 blue light. So
I agree too. I think that you need to make sure that we're consistent. You don't want to change it in the middle of the experiment, especially if it's a light experiment. You want to keep that's a variable that you want to keep the same. You don't want to change it. So, you know, so definitely for the next experiment, if you decide that you want to change the lighting, we, you know, definitely change the lighting. If you have the new system, we can help you change it. If you have the old system, you just obviously with the dials.
Yeah, and you may want to look at, you know, when you're changing it, and there might be some back and forth with Fairchild on this. So if you do a proposal for, you know, a different light treatment, you know, you want to, again, try to only change one variable for your proposal that you're putting in. So it may be, you know, like you say, the 144 to 48, but then you're kind of changing a couple different things because you're changing the ratio of red to blue, but you're also changing the overall number of photons the plants are getting.
The
So,
intensity, yeah.
yeah, the intensity. So, you know, ideally you would add up the 144 and the 72 and the 72, but those are settings. Those are not necessarily
Measurements.
micromoles. So, Amy, and Brianna, they might want to work with your team to come up with like the total micromoles, and then come up with, you know, the total micromoles from the first trial, the total number of photons hitting the plants essentially in a given period of time. And then, you know, you're shifting just the ratio of that. So it may not be the exact same settings, if that makes sense.
Basically, if you give it more light, you're going to get a different response. And if you're trying to measure the change in the color of the light, you want to give it the same amount of light. So that way you don't have two confounding variables, right? So this is what Joy said in very scientific terms. I try to
much more wordy. Yeah, exactly. You want to change either the colors or the intensity, but not both.
Yep. There you go.
Right. And I think with the proposals, like Brianna was saying, when you write your proposal and your justification for what you want to do, and if there are, and if you decide that you want to change the light setting, and that is what you've written in your proposal, when we go through and review your proposal, we would reach out to you once it's approved and everything. We would reach out to you. You could reach out to us, and we would help you. We would help figure out what that is, because like they said, you want to make sure that you're not changing too many variables by just changing the dial on the light, or, you know, something. So we want to make sure that that's correct.
All right, our next question is from, let's see. Yes. So our next question is, our average humidity in the GBE chamber is 50%. Through research, we discovered that an average humidity of 60% will most probably yield better results. We are planning to disconnect the fan for 4 hours. Would that be enough to reach the humidity we want to reach?
Once again, you don't want to do that in the middle of your experiments. So if you're planning on doing that in the experiment, if you're getting 50% humidity, that's what you're getting. And so you're in a drier location or whatever the case may be is, but you don't want to change it midstream. So if you're changing it for your proposal for the coming original research trial, then that's fine. But like if you're I would suggest, and stop me if I'm wrong, Science Peanut Gallery, you don't want to change that in the middle of your experiment. So if you're still in the middle of your 28-day trial, I wouldn't change it.
Totally agree. And the humidity on the space station is lower than 50%, right? So, you know, that's actually a really relevant target, you know, to be testing in. But those are the data that we collect, and we start to look at, wow, these plants did really good under low humidity. So they might be a good candidate for space. So that's one reason that we don't want you to change things in mid-course. But if in terms of would that be a good thing to test, that would be a really interesting thing to test for the proposal and for the science that you'll do in the spring. If that is long enough to raise the humidity, you know, we can't tell you that. It's going to depend on the humidity of your classroom. It's going to depend on how big your plants are, how much water is in your tray. So,
and temperature, don't forget.
And temperature, of course.
Temperature,
yeah.
If you turn off the fan, the temperature is going to go up.
Yeah.
Probably. And if the temperature goes up, then the air in the chamber can hold more water. So the relative humidity is actually going to probably go down, even if the total amount of water doesn't change. So
yeah,
I don't know if that makes sense, but you know, you may actually get more moisture in there, but the relative humidity may go down because the temperatures come up. So you may end up having multiple things change by doing that, and not intentional.
And there would be some things you could maybe do at the end of your experiment. After you harvest and you collect your plant weights, you still have these pots with the wet, you know, substrate in them. You could put them in there with the tray. Now, it wouldn't be perfect because you wouldn't have plants in there, right? But you could have the pots in there. You could put, you know, water in your tray. You could measure the humidity. You could turn the fan off, you know, come back in an hour, 2 hours, 3 hours, measure the humidity and see if it changed just with the pots and the water and the tray. And that might give you a feel for how long those changes might take, and also measure the temperature and see if that temperature is changing. And if you're changing, then your variable is not increased humidity. The variable, your independent variable, is turning the fan off, right? And so your dependent variables would be your humidity, your temperature, and your plant growth. These would be the things that you'd be measuring in response to the independent variable of turning the fan off. But you could do some of that before, you try it with plants and just see, does it change anything? Oh, wow, it's getting really hot in there. Well, maybe we don't want to do that. Maybe we want to think of something else.
All right, so our next question comes from Ulf Riverside. If you guys would like to go ahead and unmute and ask your question, you are more than welcome to.
Thank you. Sorry, we're having trouble with our camera. We would turn it on. Our experiment, we did the same experiment in 2 different chambers, and one of them grew that looked like luscious plants behind Joya there. And the other one grew completely out of the chamber, super tall, and it looked like we were growing something completely different. And lo and behold, we just realized a day or so ago that our timer wasn't working, so the lights were on 24/7, which explains it. So we were talking about doing our proposal where we do change the amount of time from the 12 hours to whatever to see if we could get more plant growth, dealing with that time. And we'd like to do 2 different times in our 2 different chambers. So our main question is, can we use the one that actually worked, that did the 12 hours on and off, that grew regular, as our control? And then do 2, and then just change the time so we do 2 different times in the other ones.
Yeah, I think that would be totally fine. You're lucky you've got the 2 chambers, so you could certainly do essentially 4 different, data points, and then, do a comparison. So you might have a 12-hour photo period, a 16-hour photo period, a 20-hour photo period, and 24-hour photo period, or something like that. And then that photo period, you could graph the data and just see the difference. I think that'd be really interesting.
And what's the Joy? What's the fancy term for that? Where you do the total photons added up over time?
Daily light integral,
That's
DLI. Mm-hmm.
Yeah, that's the 50-cent word, everyone. Daily light integral.
All right, thank you.
Yeah, that's cool. Plants are amazingly responsive to light, and so you can almost use them as like indicators that something's weird, right? Just like you saw, something's weird. Why are these plants growing so much differently? Everything looks the same, but then you're not there at midnight or 2 a.m. and you don't know, the lights haven't gone off. But I had that same thing happen with one of my timers failed, and the lights were just on all the time, and I was getting these weird responses, and it took me a lot of detective work to figure out, oh, okay, it was a 24-hour light period. So great that you found that.
The plants told you.
Yeah, it's wonderful you figured it out. That's awesome.
All right, so the next question comes from Gabe, who asks, we observe that the stems of our extra dwarf bok choy plants are leaning in different directions. Some of them have stem lesions and others do not. Some also have larger canopy areas than others. There does not seem to be a certain pattern of leaning. We were wondering if the stems leaning is typical for extra dwarf bok choy. Additionally, we wondered if we could do an experiment to enhance the strength of the stems and the overall health of the plants. Any suggestions for this?
Yeah, that's interesting. So I think you could do an interesting experiment on that. Now, the stem lesions is kind of an interesting thing. That could be due to some salt stress, or it could be that, the media is a bit abrasive and maybe it just scraped. And then as the plants grew, they got little scratched areas that got a little bit bigger. But the leaning, so that, tends to be due to the stems kind of elongating on plants. And so there's a really interesting property that plants have, where if you essentially touch stimulate them, that can cause them to get more short. So this is, it's called thigmotropism. It's sensing touch, and people use this in the greenhouse industry. to grow plants more compactly and to make them not lean out of the pots. They actually have, it's kind of cool. They have these booms that move across the greenhouse, maybe to spray water or whatever, and they can attach like almost like a mop or something, you know, think of like a car wash, something but small and gentle, not super heavy. And so it brushes the plants, and that will keep them shorter. And it also does what's called hardening off. So hardening off is a term where if you don't have your plants go through this and you take them from a nice, beautiful, lovely, cozy greenhouse and then you plant them out in your yard, they die or they get really stressed. So if you do what's called hardening off, and it's almost like, you know, the same idea would be like gradually moving them from this nice, wonderful environment to a less good environment, but you can get the same properties of getting resistance to stress using this type of a touch stimulation. So that's often done in the greenhouse to make plants more resistant to stress. But it also can make them shorter and more compact. So that's a lot of explanation, but the short side of it is, I think, look into hardening off, thigmotropism. We can put some of this in the chat, and touch stimulation of plants, and see if maybe brushing them at a certain age, really gently, you don't want to damage them, could help reduce that leaning. So I'll put some of those terms in the chat to look up.
All right, so our next question comes from Manuela, who says, in the radiated seeds protocol, we wonder if the radiation affects the amount of stomata beneath their leaves in the pak choi. We think to use a technique of taking out a leaf print would affect the leaves in the harvest using clear nail polish and clear tape to remove the leaf print. Then we would count the stomatas to see the differences between the irradiated seeds. We would compare the results between the trial 1 and trial 2, repeating the protocol of trial 1 because we already have the variable radiation. Let me see.
Yeah, that's great. I've used that technique. It's really cool. You may want to weigh your plants first, you know, before you do the leaf peel, because it could cause some damage to the leaves, and it, you know, so you may want to get your weights first and then do that. But I think that's a great idea.
All right, our next question is from Alexa, who asks if we have 3 different types of radishes, for example, Easter egg radish, round radish, and French breakfast radish, and some of them will do better in temperatures that differ from each other. Should we prioritize getting the best result from a specific type, or should we find a middle ground between the 3? And that is the question.
What temperature differences are you expecting? I'm not sure I'm tracking the question.
I believe they're asking if it would be better to look at prioritize the best result from a specific type of radish, or look between look for like a middle ground between all 3 types of radishes.
Can I ask a question? And Alexa, feel free to open up your mic and chime in. But is this for the first trial, or are you talking about for your original research trial?
We've already finished the original research trial, but then we're talking about for the proposal.
And how would you change the temperature in your chamber? I think that's a cool idea, but I'm curious how you plan to do it.
We were talking about maybe figuring out how to use the fan to our advantage, like turning it on more when we need it to be cooler, or changing the temperature more in our classroom.
Very interesting. It's a
Yeah,
great question.
I
Yeah,
was going to say changing the fan though would change the other parameters. So
yeah.
be very careful if you're going to change the fan setting because that changes the amount of wind that comes through, the evaporation that comes off the leaves. So there's a lot of things that change when you do that. Changing the temperature in the classroom would be good. That would certainly should impact what happens to the plant.
Yeah, you might also even just take a thermometer around to different points in the classroom, and there may just be like a hot corner or a warmer area and a cooler area, depending on, where your ventilation is. So that, moving the chamber to a different spot might be a way to do it without making everybody like super hot and sweaty in your classroom,
Or cold.
or cold. But no, I think it's a really cool question. And honestly, you could do either. You could prioritize one if you did research and you find, that certain radishes are better with, warmer temperatures, and maybe you could just focus on those. But and we have had people where they have, say all three types in the preliminary test, and then they focus just down on one type in the original research. And I think that's acceptable. You're just using, the data from that particular type of radish as your control. And then you might change the temperature. Maybe we only want, French breakfast radish or something. Or you can do all three. Finding a middle ground is not a bad idea, just because that's kind of what we have to do for spaceflight, right? We're not going to have a lot of different growth chambers. We'll probably have, someday a space like the one behind me, and you're growing a bunch of different types of plants in this one shared environment. And when we get to that point, it's going to be really, important that we have, maybe we're not optimized for any single plant, but all of them are doing good enough, right? So we're not just growing for, for onions, and we're not just growing for sweet potato, and we're not just growing for lettuce, but we're growing, that they all do okay. And maybe they're not all at their very, very best, but they're all good enough. And I think that's probably what we're going to have to do. Maybe if we're really lucky, we'll have two big chambers, and one will be a little cooler and one will be a little warmer, and we can have the crops that like it a little cooler in one, the ones that like it a little warmer in the other. But yeah, we're not sure.
Thank you.
Thank you.
All right. Well, I have a question for you too, if that would be all right. When you are usually looking to design your own experiments, what do you usually do to inspire you to make a new experiment? Like, what do you usually look at?
Well, for me, a couple different things. Sometimes I will just read the seed catalogs. I know that's kind of geeky, but there's some really neat plants out there. And so I might look at the seed catalogs and see what kind of cool plants are available. That might be a fun comparison. And, I love looking at the pictures and thinking about, wow, that'd be fun to grow, or wow, that sounds really neat. But then other things, I think about are like the problems, So the experiment that we just launched to the space station, and it's going to start growing right after the day after Thanksgiving, is based on the problem. So this is the lettuce one. I talked about it a little bit in the last chat with the scientist, and I'll be talking a lot more about it in future chats, where we're looking at different moisture levels in the substrate, in the arsenolite. And, this is because we've had countless problems, right? With not enough water, too much water, wilting, stunting because of drought, and it's always been a challenge. And so I think, well, I spent a lot of time brainstorming when we were coming up with the proposal for this project. And I was like, okay, what's our biggest problem? Well, it's, it's watering plants. We still can't get it right. It's really hard to do in space. Well, what would be interesting to study about this? Well, we're interested in how the plants are growing, but we're also interested in those plant and microbial interactions, and that's going to be really important for future systems. And we want to figure out, well, what's the best way to screw up these plant systems, right? Because we know that we're going to have failures. So if we're going to fail, well, let's try to intentionally figure out what's a good way to fail. So that's really the inspiration that I use. We do a lot of problem solving, I would say, in our job. And so figuring out, good experiments designed around problems is a great way to go.
Yeah, and I'll just say for me, when we talk about things that we need to do, I'm trying to look at the different crops that we might need for exploration and in the different families to ensure that we have biodiversity. And the biodiversity should help us with any biotic and hopefully abiotic stresses too. But and then think about increasing the number of families, but also what those might look like for meals. So I try to connect the crops, the robustness of the crops, the diversity of the crops, and then relate that back to meals. Joy has looked at herbs and things like that. We just completed the Deep Space Food Challenge a few months ago, and there was a number of chefs that I got to meet, and they're, I think, chefs and experts in the food science field will be helpful to help identify some of the herbs. Like, oh, you should really look at these herbs because you can make so many meals with them, right? And so when we look at the packaged diet and what we're growing, I look at the increasing the variety by just doing some very simple tests and decisions.
Awesome. Well, we still have a few more times for questions if anyone else has any questions. After whoa, there's some questions. All right. So, we've got one from Catherine, who asks, how would we note the amount of light exposure for the 3 varieties of radishes if each one needs a different amount of light?
Yeah, I mean, that's something that, we really can't do, right? Different amounts in the chambers very easily. I mean, in theory, you could kind of come up with a weird way to put, a piece of aluminum foil in front of some of the plants, but you'd have to do it every day, to try to block light. It's not super easy. You could use, well, that would be different intensity of light, not amount of light. So duration, like if you were trying to do intensity, you could potentially try to put a filter, like a screen or a shade under part of your light to have less total light coming to certain plants. So that'd be one way to do it, but not easy in these chambers. So I would suggest, if you want to study the amount of light as a variable, for radishes specifically, and you had 3 different types in the first test, maybe you just go down to one type in the next test and then change the amount of light, for that one, or you change the amount of light for all 3 different types, but you can only change to one, amount of light, because it makes it really hard to do that otherwise. If you had 3 chambers, you could do it, but most of us don't have 3 chambers to do that with.
All right. Our next question comes from Megan, who asks, within our experimental group, we noticed severe wrinkling in the leaves. Is it common behavior for the extra dwarf pak choi to develop this in earlier stages, and is it common for them to smooth out later on? The leaves smooth out later on.
That's a great question. Do other folks in the call have any thoughts on that? I haven't spent enough time growing the extra dwarf bok choy to really know. I think mostly the leaves kind of come out smooth, but if they're wrinkled, that's really cool.
I think that's definitely a cool note to put in your data portals and just keep an eye on those extra dwarf pak choys as they grow and keep noting that down, whether or not they're wrinkling or smoothing out or not.
Yeah, take photos, and see if they change. Now, leaves can do that. Sometimes leaves don't fully expand, or they can get like, you can get little kind of crispy edges of leaves. And maybe that would cause some wrinkling if the edges are sort of crispy and not able to expand. That can be an indication of calcium and plants just not getting enough calcium. And calcium is a really interesting element for plants because it's so critical. It's important in cell walls, among other parts of plants, but really in the cell walls, and plant cells all have cell walls. But it's not one that plants actively take up. They don't pump it up. So that means it has to come up in the transpiration, in the water that they're taking up in the roots. It's sort of dissolved in the water and it just sort of passively comes up. And the amount of water that plants take up, as you all know, is dependent on all sorts of things like airflow, the amount of humidity, and the light levels and how fast they're growing, how much water they're moving. And so, as you know, when your plants get bigger, they're using more water, right? Because they have more leaf area. So all of that brings more calcium into the plants. If you say have a fan on a plant and you turn the fan up, that'll make more water go through the plants, and that's one way to bring more calcium in. So I'm not saying to change anything now, but that could be something, you could do in a future experiment, like increase the airflow and see if it is calcium, the wrinkling might go away. But yeah, that's very interesting.
All right. Does anyone have? Ooh, that's a fun link. Does anyone else have any questions? After the session as well, if anyone does have any questions about the proposal, first trial, second trial, you're all more than welcome to email us. Let me see.
Did we
GBE
get any
at
in the form
Fairchild.
too? Didn't we have a form for questions too, Brianna? Did we get any in there or?
I don't believe we got any this time around.
Okay.
Let me double check it real quick.
Yeah, and while you're looking, I put a link to the some of the photos of the ISS pak choi, and you'll see a lot of the leaves are smooth, but some are bumpy. So depending on what you're observing, you can compare to what the astronauts grew, and see is it really bumpy compared to what they've grown. But you can see there is some little bit of variation.
All right, so we do have a question from the form. I'll go ahead and send it in the chat and read it out loud. Let me see. Our wasabi mustard plants have sprouted flowers and have little to few leaves. The flowers are also at the top of our grow box. How would this even be a practical plant on the ISS to grow since we are showing heights greater than 30 centimeters? Also very little leaves to eat. We are seeing differences in our research for what we are growing. Looks like we have wild mustard. I see our not getting the flowers online in most pictures, only looks like wild mustard. What are we growing?
Well, we have seen the same very early flowering in the wasabi. It's one that can flower really early, like 20, 21 days. It's almost more like some of the fast plants that have been grown. I'm not entirely sure the 114, but I think it probably is, what is sold commercially as the wasabi mustard. The thing about flowering in some of these is it's something that can be controlled. And so that is a really interesting thing to look up. How can you delay flowering, or how can you speed up flowering? Because that's something you can control with light, with temperature, with some other factors. A lot of plants will flower early in a warmer environment. So maybe if you are able to keep things cooler, that can delay the flowering. But you can use light, different colors of light or different durations of light also to change the flowering time in plants. So that could be a really fun project to look into.
Yeah, and it looks like they're confusing the wasabi root plant with the wasabi mustard green too. So the wasabi mustard green is one that we've grown on space station. We've grown it before. It's a pretty tough plant, when it's stressed, it will bolt. It's like, oh my God, I'm going to die. I need to get my
That's what
seeds out.
flowering. Flowering is
Yeah.
bolting. Yep.
Yeah. So, it'll flower when it's stressed because, I'm going to die. I need to get my genetics out. So, we see that sometimes, but it's a pretty tasty plant. If you like spicy leafy greens, it's one of my favorites. It's interesting to see that they didn't get very good growth. So I'd be curious to learn more about what the conditions were and why, because we have found it to be a pretty robust, reliable plant. So I would be very curious to know what was different in this case.
And you can also eat the flowers. So, even though there's not a lot of leaves, you can still eat the stems and the flowers. May not, may taste a little weird, but they should be fine. Not
Yeah,
that
I
you
don't know. I wonder if flowers would be spicy or not.
Yeah, I think they'd probably be
check
pretty good. I've eaten Mizuna flowers before. They were pretty tasty.
Yeah.
Okay. Oh, I see Lisa has a question about microbial analysis. So great
Yes.
question, Lisa. Yeah. E. coli coliforms is one that we do test for. Hopefully you won't find any of those. Those aren't really things you want on your plants, but we test for some different pathogens like E. coli, Salmonella, and Staph aureus. We have seen Staph aureus on one of our plants from the space station, Staphylococcus aureus. That's one that's commonly on skin, so I wouldn't actually be surprised if you saw that one. But it can be a pathogen. Maybe just doing general microbial testing it's good too. But yeah, it's good to check for pathogens. You can also just do aerobic plate counts or total fungi and just kind of get numbers on, how many bacteria or how many fungi are there. And those are interesting. A fun kind of spin-off project might be to get some lettuce from the grocery store or some of your leafy greens from the grocery store. If you can get something similar and compare to what you're growing in your classroom and see differences, that's always a really interesting thing. And we've done some research on that with the veggie plants and compared to what we grow in our chambers here and what we get out of the grocery store. But hopefully you won't see E. coli because those are not too good to have on your food. And hopefully you won't see anything like Salmonella either. So, but yeah, probably Staphylococcus aureus, especially if students are handling the plants, because that's one's on your skin.
All right, I believe we have time for one more question. If anyone else has a question. All right.
So I have a question.
Well,
So this will be the last chat for a little while, right? Or we don't have one in December, I don't think, or do we?
I can check on that. I do believe we.
I think we might be meeting next in January, February. Let's see.
Yes, I believe we do meet after the new year. But yeah, we should be meeting after the new year. So this is the last one for a
January
little while.
22nd, I think, is our next one. So, and that'll be on a Wednesday. So that'll be a slightly different one. So, okay. Well, wishing everybody a really happy holidays and a happy new year. So.
Yeah, and thank you, both Joya and Trent, for coming on and and answering all these questions from everyone. Yeah, I appreciate
And we
it.
really look forward to your proposals. Brianna, what is the due date for the proposals? Now, we know that some of you have had some problems getting your equipment and materials and your trial started a little late. And if you have any issues with any of the dates, you just let Brianna and the team know, and they'll make sure that, we provide you with, appropriate dates for things. But what is the due date currently for the proposal?
Yes, the due date is going to be, I believe, December 13th is the due date for the proposal.
Hello.
Hello. Did you have a question?
And Joy, Lisa had a question about how we do the microbial analysis in the chat.
Yeah, I think that's what I answered.
Okay, all right, all right.
Yeah.
I didn't know if we wanted to talk about it looked like some of the methods actually more
Oh,
so.
okay. Yeah.
Yeah, the Petri films and all that, because I think
We use
keep-
Petri films and we
Yeah.
use other plates. Yeah.
Okay.
Yeah, that would be, I think there are published methods for those. I'm not the microbiologist, but I think you mostly just, put the leaves in some buffer and then you can, take a sample of it out and stick a few drops on the Petri films and on the plates and see.
what grows.
Yeah, that's cool. Can we eat the radiated pak choi? Is it safe to eat after harvest? Oh, that's a good question.
Yeah, the radiation shouldn't impact the safety. It's really about the growing conditions and any microbial growth on the plants.
Yeah. So if anything looks funny, just don't eat it because that's our
That's
rule
what we
of
tell
thumb for the
astronauts.
astronauts. Yep. But
Yeah,
if
don't
the
eat any
plants look
funny-looking
healthy, they should be fine to eat. Yep. We can't ask you anything about the eating, but if you want to volunteer information on that, that's always of interest in the notes too. So. But we don't want to do human subject research. That requires a lot more paperwork.
All right. Well, thank you everyone for attending, and we will see you again in the new year, I believe.
Yep.
Yeah,
Happy New Year to everyone.
2025. So exciting. Tell you all about the lettuce on space station, but hopefully keep an eye out. We'll try to get some postings and get information to folks as that's growing too.
Yep.
Go team go.
All right, I'm gonna go ahead and end the Zoom so Challenge can go ahead and hop on their meeting. But yeah, it was great to see you guys.
Yep,
Good
you
to
too.
see you. Thanks, Brianna. Wonderful job. Take care. Bye,
Thank you,
everybody.
you too.
All right,
Bye,
adios.
everybody. See you soon.